Good morning, everyone, and welcome. I'm pleased to have the opportunity to present today. Today's topic is dry heat sterilization, a crucial method in our sterilization processes. My name is Dr. S.K. Singh, and I hold a PhD in this field. In this presentation, we will explore the principles behind dry heat sterilization, its applications, advantages, and some practical considerations. So, let's get started and delve into this important subject. Let's start by looking at what dry heat sterilization actually is. Essentially, it uses hot air at high temperatures without any moisture to effectively destroy microorganisms and bacterial spores. Now, you might wonder, when would we choose this method? Well, it's particularly useful when moisture could be problematic or might interfere with the material or process. For example, in depyrogenation processes, where removing endotoxins is critical, dry heat is often the preferred method. Moving on to the principle behind this sterilization, it works through oxidative destruction of cell components. What does that mean? Basically, the heat causes damage at a cellular level, leading to the breakdown of vital cell structures. This damage occurs because proteins denature, meaning they lose their structure, and the cell membranes get compromised. Together, these effects ensure that microorganisms are effectively destroyed. It's also important to note that dry heat requires higher temperatures and longer exposure times compared to moist heat sterilization. This is because moisture helps facilitate the destruction process, so without it, we need to compensate with more intense heat and longer durations. Overall, understanding these key points helps us see how dry heat sterilization fits into our broader sterilization strategies. Let's take a look at some typical dry heat cycles used in sterilization. Usually, we see cycles like 160 degrees C for about 2 hours, 170 degrees C for 1 hour, or 180 degrees C for just 30 minutes. The choice of cycle really depends on the load we're sterilizing and how we've validated the process to ensure it's effective. Now for depyrogenation, which is a more specific process to eliminate pyrogens, the temperature is higher, around 250 degrees C, and it needs to be maintained for at least 30 minutes. This ensures that any endotoxins or similar contaminants are effectively destroyed. The equipment we use for these cycles includes hot air ovens and tunnel sterilizers, sometimes called depyrogenation tunnels. These devices are specially designed to maintain consistent conditions. They feature HEPA-filtered air, which helps keep the environment clean and free of airborne contaminants. Uniform heat distribution is critical so that every part of the load reaches the necessary temperature. And controlled airflow ensures that heat is evenly spread throughout the chamber, making the process reliable and repeatable. Overall, choosing the right cycle and equipment depends on what we're sterilizing, the load size, and how we've validated the process to meet safety standards. Today, I'll walk you through the method we use during the process. First, we focus on loading the materials properly, making sure there's adequate space between items. This is crucial because proper spacing allows for effective air circulation, which is essential for uniform heating. Next comes the heating phase, where we gradually raise the temperature. It's important to increase the temperature slowly to avoid any sudden shocks that could damage the material or cause uneven heating. Once the target temperature is reached, we enter the holding phase. During this stage, we maintain the required temperature steadily. This ensures that the process is consistent and that the material undergoes the proper treatment. Finally, we move into the cooling phase. Here, controlled cooling is key to prevent contamination and to ensure the material cools evenly. Rushing this step could compromise the integrity of the process, so careful regulation is essential. Overall, each of these steps, loading, heating, holding, and cooling, are interconnected and vital for achieving optimal results while maintaining safety and quality. Let's take a closer look at biological indicators, starting with the organism used, Bacillus atrophius. This particular microorganism is highly resistant to dry heat, making it an ideal choice for testing sterilization processes. When we perform acceptance testing, we're looking for no growth after incubation, which confirms that the sterilization was effective. Now, moving on to regulatory requirements. These are crucial to ensure our sterilization processes meet international standards. We must comply with WHO GMP, EU GMP Annex 1 from 2022, and FDA guidelines. These frameworks set the expectations for validation and quality assurance. Speaking of validation, it's a key aspect here. We need to validate our sterilization processes to achieve a sterility assurance level of 10 to the -6, which means we are ensuring a very high probability that no viable microorganisms remain. Finally, there's the depurogenation requirement, which involves reducing endotoxin levels by at least 3 logs. This step is essential because endotoxins can cause adverse reactions, even if sterilization kills the microbes. Together, these points highlight how biological indicators and regulatory standards work hand in hand to ensure the safety and efficacy of sterilization processes in our industry. Now, let's take a closer look at the critical process parameters, or CPPs, that are essential for ensuring a successful sterilization process. First, temperature plays a vital role because it directly affects the efficacy of sterilization. We need to precisely control the temperature to ensure all microorganisms are effectively inactivated. Next is exposure time. The duration of the process is just as important as the temperature itself. Too short, and we might not achieve sterilization. Too long, and we could risk damaging the product or wasting resources. Airflow velocity is another critical factor. Proper airflow ensures uniform distribution of heat or sterilizing agents throughout the chamber, preventing cold spots or uneven sterilization. Speaking of chamber conditions, HEPA filtration integrity must be maintained to keep the environment sterile and free from contaminants. Beyond the CPPs, we also consider load pattern and chamber uniformity. How we load the items can influence how evenly the sterilization process works, so understanding the load pattern helps optimize results. Chamber uniformity ensures that every part of the chamber maintains consistent conditions, which is crucial for reliable sterilization. Moving on to validation requirements, these are the steps we follow to confirm that our sterilization process is effective and reproducible. Validation includes IQ, OQ, and PQ, installation qualification, operational qualification, and performance qualification, each serving a specific purpose in verifying that the equipment and process meet our standards. We also conduct heat distribution and heat penetration studies to verify that the sterilizing agent reaches all parts of the load uniformly and effectively. Biological indicator studies are used to confirm that the process can reliably inactivate resistant microorganisms. And if we are performing depurogenation, an endotoxin challenge is necessary to ensure endotoxin levels are adequately reduced. All of these factors and validation steps are interconnected and critical to maintaining a robust, compliant sterilization process that safeguards product quality and patient safety. Let's take a closer look at the advantages and disadvantages of this sterilization method. On the positive side, it's particularly suitable for moisture-sensitive materials, meaning it won't cause damage or compromise their integrity. It's also highly effective for depurogenation, which is crucial for removing endotoxins, especially in pharmaceutical and medical applications. Unlike steam sterilization, it doesn't cause corrosion, so it's gentler on glassware and metal instruments, helping them last longer and maintain their quality. However, there are some important disadvantages to consider. This process requires high temperatures, which can be a limitation for certain materials. It also involves longer cycle times, meaning it takes more time to complete sterilization compared to other methods. Plus, it's not suitable for heat-sensitive materials, plastics, or rubber items that might degrade under high heat. And finally, it's quite energy-intensive, which can impact operational costs and sustainability considerations. Understanding these pros and cons helps us choose the right sterilization method based on the materials we're working with and the overall process requirements. Now, let's take a look at some of the key applications for this process. It's used for a variety of items, starting with glass vials and ampules, which are common containers in pharmaceutical packaging. It's also applicable to metal instruments, ensuring they are sterile and safe for use. Additionally, powders like talc, which are used in many industries, benefit from this process to minimize contamination. Oils and petroleum products are also included, especially when high purity is required. One important aspect to highlight is depyrogenation, especially for glass containers before filling. Removing pyrogens, those fever-inducing substances, is critical to ensure product safety and compliance with strict regulatory standards. This step is essential in the pharmaceutical industry to prevent any risk of contamination or adverse reactions once the product is in use. So overall, this process plays a vital role in ensuring the cleanliness and safety of a wide range of products, from containers to powders and oils, supporting the integrity of the final product and safeguarding consumer health. Let's walk through this example calculation. Given that the temperature is 170 degrees Celsius and the process time is 60 minutes, we start by calculating the lethality factor. The formula here is 10 raised to the power of temperature minus 170 divided by 20. In this case, it's 10. 170 minus 170 / 20, which simplifies to 10 and that equals 1. Next, we determine the FH, or the sterilization value, by multiplying the lethality factor by the time. So, 1 times 60 minutes gives us a total of 60 minutes. This means the process meets the sterilization requirement. Now, for depyrogenation, the goal is to destroy endotoxins. A typical example is heating at 250 degrees Celsius for 30 minutes. This temperature and time combination is effective to ensure endotoxin destruction, which is crucial for certain sterile applications. Overall, these calculations help us verify that our sterilization processes are effective and meet safety standards. Now, to wrap up, let's look at the key takeaways about dry heat sterilization. This method is highly reliable because it uses high-temperature hot air to sterilize, primarily through oxidative destruction of microorganisms. It's especially important when we need to depyrogenate glassware and other heat-stable materials. The process relies on controlled heating, ensuring that the air circulates uniformly around the items, and that the exposure time is validated to guarantee complete sterilization, all in line with GMP and WHO guidelines. One of the biggest advantages is that it's suitable for moisture-sensitive materials, which can't handle steam or liquids. It's also effective at removing endotoxins and doesn't pose a risk of corrosion to the equipment. However, there are some downsides. It requires higher temperatures and longer cycle times compared to other methods. Plus, it's not suitable for heat-sensitive materials like plastics and rubber, which could be damaged by the heat. Overall, when properly validated and controlled, dry heat sterilization becomes an essential tool in pharmaceutical settings, especially for applications where moisture can't be used and controlling pyrogens is critical. It's all about choosing the right method for the right material to ensure safety and efficacy. For further support, please don't hesitate to reach out to us. We're here to help you with any questions or additional information you might need. Remember, your success is our priority, so feel free to contact us at any time. Thank you so much for your time and attention today. We appreciate your engagement and are excited to see how you apply what you've learned. Again, thank you and have a great day.